HMP shunt is an alternative pathway for oxidation of sugars via glycolysis and TCA cycle. Actually, in most animal tissues, the major fate of glucose is by glycolytic breakdown. But if the cell needs some specialized products like riboses, this HMP shunt operates. The HMP shunt operates in the tissues which actively synthesizes fatty acids and steroids such as liver, adipose tissue, mammary glands, adrenal cortex, etc. This is also known as pentose phosphate pathway. The main purpose or significance of the pathway is production of NADPH for biosynthetic reactions of fatty acids and production of pentoses for nucleic acids. So let us see what is the importance of NADPH in our body. They provide reducing power for biosynthetic reactions. They are also required for synthesis of fatty acids, cholesterol in liver and adipose tissues. Also, NADPH are needed for amino acid biosynthesis involving glutamate dehydrogenase. And NADPH prevent toxicity of H2O2, which is produced continuously in our body. NADPH are involved in detoxification of drugs. It is required for phagocytosis. In particularly RBC and corneal cells, the role of NADPH is very special and it protects the corneal cells and RBC by the oxidative reactions of nascent oxygen. So, these pentoses which are produced by HMP shunt are useful for synthesis of DNA and RNA, and also they are useful in the synthesis of coenzymes like ATP, NADP and FADH2. HMP shunt is rather considered as anabolic pathway because it is involving in the production of pentoses and this NADPH. No ATP is directly utilized in HMP pathway. Enzymes of HMP shunt are located in cytosol of liver and adipose tissue. HMP shunt starts with 6 molecules of glucose 6-phosphate and at the end 5 molecules of glucose 6-phosphate are recovered and regenerated and 6 carbon dioxide are produced. So the pathway is divided into oxidative and non-oxidative phases. In oxidative phase of HMP shunt, which is an irreversible phase, glucose 6-phosphate degrades into ribose 5-phosphate and NADPH. In non-oxidative phase, which is reversible phase, from this ribose 5-phosphate, again glucose 6-phosphate molecules are regenerated. So this ends the pathway.